In this video class, we are going to learn one more type of gene interaction. In the beginning of this chapter, we studied about the gene interactions are two types. It is called as allelic interaction and non-allelic interaction. In the case of allelic interaction, we studied incomplete dominance, codominance, lethal genes, isn't it? Now we are going to continue about non-allelic interaction, which is also called as intergeneric gene interaction. Intergenic interaction. In a chromosome, genes are arranged in a linear order. The place where the gene is located is called locus, plural form loci. The pair of alleles which are present in the same locus of a chromosome or may be found in different chromosome where their interaction will be studied. Interlocus interaction takes place between the alleles at different loci. Interlocus interaction between the two locus. The interactions takes place between the alleles at different loci, namely between the alleles of different genes. It is not takes place within the gene alleles of a gene. It is takes place between the alleles of different gene. So different genes, that means it may be present in same chromosome in different loci or different chromosome in various locus. So alleles of different genes, when they interact, which is called as intergenic gene interaction. In the previous class, where we studied intragenic or allelic interaction, alleles of the same gene when they interact, incomplete dominance, mirabilis jalaba, in which we studied about incomplete dominance. It is also called blending inheritance. The phenotypic ratio, genotypic ratio is same. It was first studied in 4 o'clock plant called mirabilis jalapa, in which color of the flower is not dominant over one another. Its phenotypic ratio, genotypic ratio in the F2 generation is 1:2:1, and also it is studied in Snapdragon Antirrhinum majors. The same phenotypic ratio 1:2:1 was obtained. Codominance also we studied, in which cotton plants. In human being, it is ABO blood group system, in which the phenotypic ratio is 1:2:1. But intergeneric or non-allelic interaction, which takes place in different. So between the alleles of two different genes present in the same chromosome or in different chromosome, the interaction is taking place. Based on the nature, intragenic interactions, in which several types were studied. We are going to study dominant epistasis in fruit color, of summer squash. We are going to study, and also many other types are given in the table. You can see the fruit color in summer squash, recessive epistasis, flower color of Antirrhinum species, duplicate genes with cumulative effect, fruit shape in summer squash, it's nothing but cucumber. And the next, complementary gene interaction, flower color in sweet peas, it is called Lathyrus odoratus, studied by William Betson and Reginald Punnett in this plant. If you observe where the mental diabetic cross ratio, what is the diabetic cross ratio? 9:3:3:1, but it was not that. 16 phenotypes, 16 individuals are produced, and the phenotypic ratio of the diabetic process 9:3:3:1. Here the 9:3:3:1 is modified into different phenotypic ratio. Supplementary gene interaction studied in color of maize 9:3:4. Inhibitory genes and duplicate genes in plants were studied. Okay, so the phenotypic ratio of the various epistatic interaction in intergenic interaction was given. So what is the benefit? You can understand how the alleles present in the different loci of the different genes are interacting with one another, how they are changing the phenotype or how they are affecting the phenotype of an organism, and how it changes the phenotypic ratio of the mental diabetic cross. That we are going to study in detail. Okay, children. Now continue intergenic interaction. Intergenic interaction, it includes dominant epistasis, recessive epistasis, duplicate genes with the cumulative effect. First we are going to see dominant epistasis. In dominant epistasis, where one gene which masks or inhibit the effect of another gene. You know what is called dominancy. Mendel, when he conduct monotypic class, he chose one pair of contrasting character at a time. When he cross a single trait, a single gene which controls a character, it has two alternative or distinct traits. A character which have two distinct traits, namely stem height in which tall and dwarf. What was the F1 generation shows? All were tall. So the tall character is called a dominant character, and the character which is masked is called a recessive. Dwarf is recessive, that we know. So the alleles for the dwarfness called small t, when they are in homozygous condition, it produces dwarf. If they are in heterozygous condition with the dominant gene, that is capital T, small t, dwarf character will not be produced, because the dominant allele capital T will inhibit or mask the effect of dwarf. When the F1 generation is crossed by self-fertilization, it produces both the parental phenotype, that is called tall and dwarf, in F1, F2 generation as 3:1 ratio, that we knew thoroughly. But here if you observe, dominant gene and recessive gene is there. Apart from that, due to the presence of an epistatic gene which control the effect of other gene. So due to the presence of epistasis condition, one gene which masks or inhibit the effect of another gene. This epistatic gene found in different loci of different gene. So interaction between the alleles of two different genes is called what? Intergenic interaction. Let us start the definition. Dominant epistasis. It is a gene interaction in which two alleles of a gene at one locus interfere and suppress the mask of the phenotype expressed of a different pair of alleles of another gene at another locus. Very simple. One gene which have a pair of allele found in one locus, another different gene found in another locus, in different locus, having alleles. So alleles of one gene found in the one loci which suppress or mask the phenotypic expression of different pair of alleles of another gene at another locus is called a dominant epistasis. In this case, the gene which is suppressed at another locus is called epistatic gene. So one locus, a pair of allele, belongs to a gene which affects or suppresses the phenotypic expression of different pair of alleles of another gene that found in another locus. So the gene which suppresses the effect of another gene is called epistatic gene, and the condition is called epistasis. So the gene that suppresses or masks the phenotypic expression of another gene which found in another locus. So gene that suppresses or masks the phenotypic expression of another gene in a locus is called epistatic. This gene, the gene whose expression is interfered by non-allelic genes which prevent the exhibition of their character is called hypostatic character. Simply, if you say the gene which suppresses the phenotypic effect of another gene which found in a locus is called epistatic gene. The gene which was suppressed and its phenotypic character was suppressed, the suppressed gene is known as hypostatic gene. Hypostatic gene always suppressed by epistatic gene. So epi means superior, hypo means inferior. Keep remember. So in this epistatic epistasis, a pair of alleles of a gene present in a locus which affects the phenotype of another pair of alleles of a gene in the different loci is called epistasis. The gene which masks the effect of phenotype of another organ is another gene is called as gene in a locus is called as epistatic gene. The gene which suppressed its phenotype, the gene whose phenotype is suppressed, the particular gene is called a hypostatic gene. When both the genes are present together, the phenotype is determined by epistatic gene, not the hypostatic gene. In this epistasis, dominant epistasis was studied first in summer squash plant, in which color of the fruit. There are three types of color of the fruits was observed in this summer squash, that is called white color fruit, yellow color fruit and green color fruit. Due to the presence of the epistatic gene, the color of the fruit was determined. In summer squash plant, for example, the summer squash plant, here you can see the slide, dominant epistasis. In the summer squash plant, the fruit color locus has a dominant allele that is called capital W. There are three colors in the fruit of summer squash was studied in dominant epistasis. It was controlled by the particular gene. Here the epistatic gene is capital W, which produce the white color. It's a dominant allele, which the dominant allele W for which the white color, and the recessive allele is small w for the color of the fruit. If the dominant allele, that is called epistatic allele, that masks the expression of any color, it does mean what? If the epistatic allele which is dominant called a capital W is present in genotype of any summer squash plant, its color will be always white. Always white. The yellow and green color of the fruit will be suppressed. Okay. In another locus, the hypostatic allele called capital G is present. The capital G, the hypostatic allele, will not produce yellow color if the epistatic gene W is present. So the dominant allele W, which is epistatic, which does not allow the expression of hypostatic gene, that is called G. So hypostatic allele G will not produce the color called yellow, okay, the color called yellow, if the dominant allele W is present. And the double recessive gene is called as small g and small w. So the recessive allele G, which produce the green color fruit, if both the epistatic gene called W as well as a hypostatic gene called hypostatic allele called G, both were absent. So hypostatic gene is not present, epistatic gene is not present, then only the recessive color of the fruit called green will be produced. Green will be produced. So in the first locus, white color is dominant to the color, whereas the second locus, yellow color is dominant to the green. When the white fruit with the genotype of W, small g is crossed with the yellow fruit of small w with the capital G, all F1 generation fruits color will be what? F1 generation in which the heterozygous, the fruit color of white will be produced. So you can see the cross. You can see the cross. The cross in which you can observe capital W, small g. The cross between the two parents. One parent which produce the white fruit, its genotype is capital W, small g, and yellow color fruit which produced by small w, capital G. The dominant allele capital W is called epistatic gene, okay, and the hypostatic allele is called capital G. If both are not present, keep remember children, if both the epistatic gene and the hypostatic gene is not present, in the recessive state, the color of the fruit will be what? Green. So the first locus in the chromosome, color for the white dominant allele is present, that is called W. The second locus, the color for the yellow color fruit called hypostatic allele capital G is present. In another locus where the white color fruit producing, the green color fruit producing recessive gene is present. So if the F1 generation, when the parents of white color fruit and yellow color fruit with the genotype of capital W, small g, and small w, capital G, when it is crossed, the F1 generation all were white color fruit. You may ask question, sir, capital W, its genotype if you observe, capital W, small w, capital G, small g is there. Both the hypostatic gene is there, epistatic gene is there. Why the white color fruit alone produce, sir? Keep remember, to produce the white color in fruit, in summer squash, the dominant allele called capital W, only one is enough. So only one dominant allele capital W is present. Definitely the color of the fruit will be white, since it is a epistatic gene. So the phenotype of the F1 generation is the fruit color is always white because of the dominant allele which is epistatic called capital W. All were white color fruits. F1 generations are self-fertilized. They produce 4 different types of gametes. capital W, capital G, capital W, small g, small w, capital G, small w, small g. Keep remember epistatic gene is capital W. If one capital W is there, the fruit color will be always white because it masks the effect of yellow color as well as the green color of the fruit. If capital W is absent, the hypostatic gene or the hypostatic allele capital G is present. Due to the absence of epistatic gene capital W and the hypostatic allele called capital G is present, the color of the fruit will be yellow. It won't be green. Third condition, if both the epistatic gene or epistatic allele called capital W and the hypostatic allele called capital G both are absent, in the double recessive condition, the color of the fruit will be green. Its genotype is small w, small w, small g, small g. Now you see the checkerboard. In this checkerboard, where it is very clear, F2 generation are produced in the phenotypic ratio of 12:3:1, where white color fruits are 12, yellow color fruits are 3. The double recessive fruit color of the summer squash green is appeared as 1. It is the modified genotype, modified phenotypic ratio of mental diabetic class called 9:3:3:1. So, in this checkerboard, if you observe, wherever you see white color of the fruit, in which at least one dominant allele called capital W will be there. Either one allele or two allele is present, definitely the color of the fruit will be white. So, you can see 12 white color fruits in phenotype. And the condition number 2, as I told, the color of the fruit is, if it is yellow, definitely epistatic gene called capital W will not be there. So if you see the yellow color fruit in this genotype, capital W gene is not there. Instead, hypostatic gene called capital G is present. So if at least one capital G is present, in the absence of epistatic gene, the color of the fruit will be yellow. In the case of green, both the epistatic and hypostatic gene is absent. So in the double recessive state, small w and small g which produce the green color fruit. So the phenotypic ratio of the dominant epistasis is 9:3:1. 9:3:1. Okay, children. Now see the condition. Page number 45 in your book. Since the W is epistatic to alleles of capital G and alleles of small g, white which is dominant masks the effect of yellow or green. The homozygous recessive of small w genotypes only can give color fruits 4/16 out of 16. Double recessive small w small g will give the green fruit 1/16. The plant which having only capital G, the genotype will be either small w capital G small g, or small w capital G which give the yellow color. So what was the change? You can observe the color of the fruits which appeared in the F2 generation. They produce the phenotypic ratio as 12 white, 3 yellow and 1 green. Here the epistatic gene is capital W. Either in the homozygous condition or in the heterozygous condition, it produces only white color in the fruit. In hypostatic gene, yellow color will be produced due to the presence of capital G and the absence of the epistatic gene capital W. In the double recessive cases, small w small g will produce the green color, instead the white and yellow. May hope that you are followed the explanation for the dominant epistasis. Go through carefully, children. It's very important. The phenotypic ratio can be asked in one word question. The definition can be asked as dominant epistasis as 2 mark question.